So a lot of times with cloning, obviously there's big ethical issues. We can't just be cloning ourselves, but that doesn't mean we'd all be exactly the same, because a lot of how we are and what makes us who we are is determined by nature. So you always hear the nurture versus nature. So nature would be like your genetic disposition, and then your nurture comes from your environment. So a lot of what makes us who we are is an environmental factor as well. So anyway, embryologic cells are obviously cells of embryos, and they're not like a certain type of cell yet. So after the sperm and egg fertilize, we get this huge mass of cells for about the next week, and they haven't really become any type of cell in particular. They're just genetically identical copies of each other, because that sperm and egg fuse together to create one cell, and then it divides through the process of mitosis until we create the entire embryo, and then that happens through the rest of your life. So all of your cells from your body stemmed from the fusion of that one sperm and egg together. So they haven't differentiated, meaning they haven't become the type of cell that they're going to become yet. So we hear a lot of cloning in terms of using embryologic cells. And there is some truth to that. It's not so much what is said on the news. It goes way beyond the scope of what we need to know here. But obviously, for the very, very, very early stages of the development of an embryo, all of the cells are undifferentiated. So the idea behind using embryologic cells or stem cells is that we can guide their differentiation and hopefully make new cells of a certain type. So like this type of technology using stem cells would be able to essentially regenerate spinal tissue for somebody who might be paralyzed. So that's kind of the idea of where they're going with that, but there has been little to no success in actually doing that. And so there are many other places, like we all have stem cells in our bodies still. So there are adult stem cells, and there are embryologic stem cells. So they are very different, and we've actually had a lot more success making different types of cells and tissues using adult stem cells. So obviously there is a whole lot of controversy with using embryologic stem cells, but again, it is strictly like in a lab setting, and they're not actually taking like fetal tissue cells. And again, it goes like way beyond our scope here. So basically, when we are trying to clone something, we want to make an exact replica. So what we have here, down here, this big cell is an egg cell. So it's a mature egg cell. And we have to get that, and we know the egg cell from the mom has half of the DNA of the total organism, because you know that you get half from mom and half from dad in terms of your DNA. So this little dot right here represents a nucleus in there that contains the DNA that you would get from your mom. So in this egg cell, we basically take this nucleus out. So we enucleate this cell, and we take a somatic cell, so any other type of body cell from the same organism. We take a somatic cell that we know has a full set of DNA, so basically like a skin sample we could take, and we take the nucleus out of that somatic cell, and we insert it into the egg cell that we've taken the nucleus out of there. And it, over time, obviously there's a lot more stuff that goes into it in terms of chemicals and growth factors and enzymes and proteins that need to be there. But with all of the right conditions and the right environment, this will then develop into an exact replica or a clone of the organism where we got the somatic cell from. So basically the DNA from this somatic cell is the DNA that's in here, which is in all of the cells of this new organism. So that's how cloning occurs. Obviously, it's a much more detailed process than that. And we've done this with multiple different animals. We've done it with mice, we've done it with sheep, we've done it with monkeys, all sorts of different animals, probably way more than we know about. But it's been really, really, really limited success. So Dolly the sheep was the first one, and it was somewhere over in Europe, I think in Scotland maybe, where they cloned the first sheep, and so Dolly was the first sheep. Certain things that happen with cloned organisms, though, is that they do not survive for very long. So they have a really, really, really short lifespan, and scientists aren't sure why we see that happening with the clone and not the regular organism where we got the donor nucleus from the somatic cell from. We're not really quite sure why that happens. So That's essentially what is being shown here. So the fused cell, here's my egg with the nucleus taken out. We've taken the nucleus from right here from a sheep's udder, it says, and so we've taken the nucleus and inserted it here. This is the fused cell, the egg with the nucleus from a somatic cell in that sheep. And so then it starts to develop into the embryo. The embryo is placed in the uterus of a foster mother, so kind of like in vitro type fertilization, and then the embryo develops normally into a lamb, and you can see Dolly was the first cloned one here, is the example they're giving you. All right, so again, just another picture of it. So pros and cons of cloning: we can save endangered species. We might even be able to bring back some, like if you think about Jurassic Park, it's that kind of idea. So we could bring back some extinct species if we had the DNA from said species. We'd also need an egg cell, so maybe that wouldn't probably work. But so we could save endangered species by creating duplicates or replicas and hopefully putting them back in the wild so they have more in their population to reproduce. Transgenic animals for human consumption, so again for our purposes only, for our betterment. And then we have to think about organ and tissue transplants as well. If I could grow a whole another kind of organism that would just be used for parts, then that's kind of what this is going for. Obviously big ethical issues here. Cons that we've come across: cloned animals have lots of genetic defects. Again, we're not really quite sure why, because the animal where the nucleus came from didn't have these genetic defects, but we do see them. And so with that, we see a slew of health problems, and then we have a very, very, very short life expectancy. So these organisms tend to die really young. So just some of the different applications that we can use DNA technology for, and that we have. So it goes well beyond our scope. We have an entire pathway on this stuff because it is really, really, really cool. But for our purposes, these are the things that we need to focus on. So based on what you have noted on the bottom of your sheet, I want you to answer this question. So your guided notes should be complete. This PowerPoint and all of these are posted on Schoology, but how did we actually get this pig's snout to glow? I want you to kind of outline in 5 or 6 sentences. How did we actually get these pig snouts to glow? Okay.